Very good evening, students. Most welcome, very warm welcome to everyone for today's session. I hope you are all doing great. Looking forward to our first lecture of today, where we'll be dealing with the basics of cell biology and molecular biology. Because some of you might be in advanced studies, some of you might be at the back part of your studies. So I'm just teaching for the students who never had anything about cell or anything known about molecular biology. Before we start with techniques, we go a little bit about the basics today. So that's what we're gonna start with. Anyone has questions from previous session? Any about from review assignments? All was clear. I got so many also questions on Telegram, so I tried to reply them. So I hope connection is fine so far today. There's no lagging is happening. So we will see how far we could go with this such a great internet today. Um, I wait for 4 minutes, 2 or 3 minutes more, then we start with the session. Yes, yes, Neha, you can choose any specific branch within the cancer cell research. It's up to you how you want to look for. It's all open. Good evening, good evening. So it's 7:06 now. We will start at 7:08 or 7:10. Thank you everyone who has subscribed to my channel. I really appreciate that. It has raised to high level now. So Somdata Mighty, it's not necessary that you have to submit assignments every day, but if you do so, we will be very happy. It's good for your own practice. Okay, Dia, you can write it in the doc, but in the future, you will have to make all documents in doc. So it's better you start preparing for them also from today onwards. So there is Bhavya Gupta, there is no limit for review page limit. You can write as much as you can, but at least you should have 50 references. I repeat, at least 50 references to make it a really good review article. But its assignment is just you have to write a summary actually, Rashi. You have to write summary of each topic that we will be dealing each day, and you have to write a very nice a little bit summary about it. Or review should be digital, no, not handwritten. Of course. Of course, Neha, it has to be digital. Yeah, yeah, Nishita, everyday classwork. That would be your assignment. So you can send it. I have already shared yesterday my email ID and info at the rate of edufabrica.net. You can send it on both. So please pin my messages so that you can come to know because my messages are lost in your chatting actually. So I have shared my email ID. If you go through the topmost part of the conversation, you can see it from there. No, if one can submit, that's okay. But in order to receive your articles, individual will be also fine if you can do it. Okay, students, we have talked a lot now. Let's start the session. It will be late otherwise. So you might be aware that cell is like a factory. So I made this analogy here where we'll be dealing how each part of a cell from your mitochondria to a nucleus to chloroplast, nucleolus, nucleus, all how they're working with each other in a very nice manner. So let's start with this analogy here. So how can a cell be act like a factory? So in a factory, you have many different tasks being completed in the same building, like shelters, workers building goods, maintenance of crews, cleaning messes, shipping departments. All these being dealt in while doing working in the factory. And cell could be act as an analogy as a factory where the same thing are being specialized with the different departments, such as, let's start from the cell wall, which acts like a security gate. So security gate protects a factory and helps keep it safe. A cell wall does the same thing for a plant cell. So here you can see. So security gate protects the factory and helps keeps it safe. And cell wall does the same thing for the plant cell. And then comes the cell membrane, which acts like a front doors. So doors of a factory regulates what enters and leaves the building. Cell membrane does the same thing um, for the plants and animal cells. So here you can see the doors which let to enter anything or should not be. And the nucleus acts like a big boss CEO which controls the activity of the cells for the both plant and animal cells. Where the cytoplasm is like a factory floor, where the floor of factory has all the worker machines and equipment on it, and cytoplasm holds all the organelles in the plants and animal cells. Whereas your ER, endoplasmic reticulum, acts like a assembly line where the workers do their work, items move along the conveyor belt to different parts of the plant. So they are moving things from one place to another. And then ribosomes here, they works on the assembly line. They are responsible for building the goods in a factory. A ribosome assembles build proteins like this. Then vacuoles are the break rooms where you have storage. You have to store food items. Does not need get rid of the trash. So vacuoles hold waste, stores water in plant and animal cells. So which cell has one large vacuole? So here you can see. So lysosomes are like maintenance janitors crews in a factory. These guys break down, clean up and repair items in and around the factory. Our lysosomes do the same things in the cell. Then mitochondria and chloroplasts, they act like power machines in the factory. And the mitochondria and chloroplasts do the same for the cell. So this is a whole example of a cell where your ribosome acting as assembly line here, mitochondria as a power cell, yeah, endoplasmic reticulum as assembly line, nucleus as a powerhouse of the cell. There's some restricted areas, the cytoplasm. Then there's a Golgi apparatus getting the waste products. Then the proteins production is being done. So this is how your complete production of your proteins are being done with the help of your cell. So that's it. So that's something very basics. Um, Pranav Rana, okay. But for those the screen is not visible, try to increase its uh, your what do you say your resolutions to 480p. Only then you can have it in a better way. Pranav Rana, sorry, I cannot share them again. Please ask Vatsal sir to share with you. Those who have told you that you will get things done, so please ask them. Okay, so that was a very basics about cell biology. So as soon as we finish one topic, so I will share that topic with you within the group. Okay, it's being uploaded. So all is good so far. Nothing breaks out. Or students, please share, help Pranav Rana so that he could also have the notes again. Share them or forward them in the group so that it could be assessed to the new students also. Okay, so next part, basics of molecular biology. So we had discussed so far what is cell is, how cell is working, what are the various parts of it. Now it comes to how the cell is working, like what is DNA and RNA, how protein production is done, what is transcription, translation. So that what we're gonna deal with. I hope 90% of you might be knowing about all this. I know, but still for those who are not familiar with these things and want to have a strong base first, then we start with the techniques. So I that's why I keep these topics always in the start of my MBBT course. So let's start about the basics of molecular biology first. So there are basically two types of your cells or organisms, that is eukaryotes and prokaryotes. And eukaryotes includes your plants, animals, and fungi, whereas prokaryotes includes your bacteria and cyanobacteria. And the cell wall is not present in eukaryotes, whereas in prokaryotes it's present. And further, meiosis is happening in eukaryotes, not happening in prokaryotes. And all the membrane organelle cells are present in the eukaryotes, whereas the cell organelles in the prokaryotes are absent, such as nuclear membrane, ER, Golgi apparatus, mitochondria, chloroplast. So molecular biology is a study of molecular underpinning of process of replications, transcription, and translations of genetic material. So this field of molecular biology, it includes animals, cells, plants, microorganisms, molecules, and populations. So, so here you can see that there is animal development is being correlated with animals and cells, molecular neuroscience to be interconnected with animals and cells also, and that's how all things are being interconnected with each other. So this field overlaps with other areas of biology and chemistry, particularly genetics and biochemistry. Molecular biology chiefly concerns itself with understanding the interaction between the various systems of cells, including the interactions between DNA, RNA, and protein biosynthesis, as well as learning how these interactions are regulated. So much of the work in molecular biology is quantitative, and recently much work has been done at the interference of molecular biology and computer science. And your recently DNA was found in the 1950s, then reciprocally your RNA and all other molecules are being discovered actually by the molecular biologists. So the main components that involved in the molecular biologies are your DNA, RNA, and protein. So genetic unit of heredity, gene is a genetic unit of heredity. Why it is called as genetic unit of heredity or genes? because the DNA segments that carry the genetic informations, they are called as genes, and it is normally stretch of DNA that codes for type of proteins and functions of organisms. Here you can see that there is a cell. Within the cell, you have these chromosomes, yeah, with a centromere and the satellites being up and down, and they are being and within them they are being called by the histones, and your DNA is being coiled around this like this, and then it's a helical structure having basically made of four different uh, nucleotides, that is A, T, C, G. That's the basic foundation of your gene is made up of. So actually DNA is a nucleic acid that contains genetic instruction used in development and functioning of all living organisms and some viruses. So DNA is a set of blueprints needed to construct other components of cells such as proteins and RNA molecules. So two long strand makes the shape of double helix. So here you can see this G will make with the C, A will make with the T, with the A and T is making double bond, and whereas G and C is making triple bond. And these bonds are mainly your hydrogen bonds, and on the side you can see very nice parallel sugar and phosphate backbone chain, and that distance of one nucleotide pair is around 3.4 nanometer, and the radius is around 1 nanometer, and the distance between two nucleotide pairs is around 0.34 nanometers. And that's how the helical structures looks like. And the two strands, they run in the anti-parallel way, with the 5 prime to 3 prime on one end and 3 prime to 5 prime on the another end. So chemically, your DNA consists of two long polymers of simple units called nucleotides with backbones made up of base, sugars, and phosphate. The sugar is made with the base, then it's known as nucleoside. Bache, is the screen is being shared to everyone? Why somebody saying, Vishal is saying that screen is black, presentation is not available, presentable? So I just came back to check if everything is fine. Is screen being visible and my voice is okay so far? Please reply to it. Yes, it is visible. That's good. So let's continue. And then if your phosphate is being added to sugar and base, then it's known as nucleotide. So that's the basic difference between your nucleotide and nucleoside. So they are bases. So they are adenine and guanine. So your adenine is made up of this much of base pairs. You can see one is six ring and one is five ring, and together they make this chain. And then the guanine on the other side. And on the other side here is your cytosine, thymine, and uracil. And this is how your structure is made. Whereas the cytosine and thymine is a six-membered rings, they are called as pyrimidines. Whereas adenine and guanines, they are called as purines. A fifth pyrimidine base called uracil usually takes the place in case of RNA. So in RNA, instead of T, you have U present. So all is good. That's good. My voice goes to you later, so that's why I get reply late. Understood, understood. Then your DNA is a double helix and stabilized by the hydrogen bonds. Here we have discussed it already. Adenine and thymine making a double strand, whereas guanine and cytosine making a triple strand, triple bond hydrogen bonds. And the main difference between your ribose sugars and deoxyribose sugar, that is your DNA and RNA, is of the second carbon. You have this hydroxyl group absent in the DNA, whereas in the RNA it's present here on the second one. and the rest is the same pentose sugar without hydroxyl group. This is your, yeah, the DNA chain is around 22 to 26 angstrom wide and have one nucleotide unit that is 3.3 angstrom long in this case. So ribonucleic acid RNA is a biologically important type of molecule that consists of long chain of nucleotide units. They have nitrogenous base, ribose, sugar, and phosphate. It might be not all helical, it might be a little bit helical here, but most of the time it's also single stranded. It also have base pairs, but sometimes, but always it will, instead of T, it will have uracil in this case. So double-stranded RNA is RNA with two complementary strands similar to the DNA found in all cells. So double-stranded RNA forms the genetic materials of some viruses. So here you can see HIV virus, it will have protein coat outside and the RNA genetic material will be inside of it. Then there are different types of RNAs, that is your mRNA, ribosomal RNA, and transfer RNA. Whereas your mRNA codes for proteins, whereas rRNA, ribosomal RNA for translation, whereas a tRNA for the translation. And in the post-transcriptional modifications, you have also a small nuclear RNA, yRNA, telomerase RNA. And you have regulatory RNAs as antisense RNAs, which has transcriptional attenuations and translations blocks attached to it. Yeah, all good. Yeah, there I can see that there are mainly 98 students left here. That's a good point, Venkatacharan. That we can shift to Google Meet for the next classes. Because for me also YouTube is consuming a lot of data here, and it's not a very well way to go for at the moment. But anyhow, as the students percentage decreases, we will shift to Google, but not at the moment because it will be problem for me then. But for the moment we will continue like this. As students get less, so there are 100 students now, okay. So so that's the thing. Anyhow, let's continue. So messenger RNA carries information actually from your about protein sequence to the ribosomes and the protein synthesis factors in the cells. So it is a, so like here you can see you have a coding strand here, and this will have a messenger RNA, and then you have G U U A C C G A U, and then messenger RNA will be it is coded so that every three nucleotides corresponds to one amino acids here. So in eukaryotic cells, one precursor mRNA has been transcribed from DNA and it is processed to mature mRNA. And this removes the introns, non-coding sections of your pre-mRNA afterwards. And the mRNA is then exported from a nucleus to cytoplasm where it is bonded to the ribosomes and translated into its corresponding protein with the help of tRNA. So in prokaryote cells which do not have nucleus and cytoplasm compartments, mRNA can bind to ribosomes while it is being transcribed from DNA. Then transfer RNA, tRNA, it's a small RNA chain of about 80 nucleotides that transfer the specific amino acids to the growing polypeptide chains at the ribosomal site of protein synthesis during the translation. So here it will have your codons at the bottom where messenger RNA will be running and anticodons on top. You will have intramolecular base pairing here and also ester bonds at the end. So this is how your tRNA specifically looks like. Then there is a ribosomal RNA. So ribosomal RNA is a catalytic component of the ribosomes. They contain 4 different ribosomal RNAs, that is 18S, 5.8S, 28S, and 5S RNA. So RNA molecules are synthesized in the nucleolus. So in the cytoplasm, ribosomal RNA and protein combines to form a nucleoprotein called ribosome. So rRNA is extremely abundant and makes 80% of the 10 milligram per ml RNA found in typical eukaryotic cytoplasm. So what is the main difference between your DNA and RNA? As they contains ribose sugars, base is uracil, presence of hydroxyl group at the second position of ribose sugar, and RNA is usually single stranded. Another case is double stranded. And protein, also known as polypeptides, are made up of amino acids arranged in a linear chain and folded into a globular form. The sequence of amino acids in a protein is defined by the sequence of a gene which is encoded in the genetic code. And genetic code specifies around 20 standard amino acids. So here you can see various amino acids being chained together, having a disulfide bonds between them, are making a specific structure, then like a tertiary structure, quaternary structure, and we can have it in the form of eggs, meat, fish, pulses, nuts, legumes, your what are they called in English? Chanas. Uh, I forgot the name. So all these, and then how they are playing role in the molecular biology. So first is your DNA, then comes your RNA, then comes your protein. So production from DNA to RNA is called as transcription. From RNA to protein it's called as translation. And DNA could also self-replicate, and this whole process is known as central dogma of life. So DNA replication is the basis of biological interference. It's the fundamental process in organisms to copy our DNA. So two identical DNA molecules have been produced from single double-stranded molecules. So here you can see in a cell, DNA replications begins at specific locations in genomes called origins. So you will have a topoisomerase which will unwind your DNA, and the helicase will start to unwind it, and you will start to have single stranded binding proteins. Then you will have one lagging strand and leading strand. And the leading strand, you will see the DNA polymerase is being attached there, and will keep producing your double chain molecules. Whereas in the lagging strands, you have still DNA polymerase, but you will have some Okazaki fragments, which will be filled with the help of DNA primase and RNA primase. And that's how your DNA replication is done at the end. Then comes your transcription. Transcription is the process of creating an equivalent RNA copy of sequence of DNA. So transcription is the first step leading to gene expression. So here you can see DNA is producing your RNA. It's known as transcription, but could also go reverse. Then it's called as reverse transcription. So during transcription, DNA sequence is read by RNA polymerase, which produces a complementary antiparallel RNA, and transcription results in RNA complement that includes uracil instead of thymine. Then transcription process, the stretch of DNA transcribed into an RNA molecule is called transcription unit and encodes at least one gene. If the gene transcribed encodes for proteins, the results of transcription is messenger RNA. And this messenger RNA will be used to create that protein by the process of translation. So alternatively, the transcribed genes may be encoded for either RNA or tRNA, other components of protein assembly process, or other ribosomes. So this is how you can see your RNA polymerase is being attached there to your promoter sequence. Then it's producing at the terminal signals. At then you will have proper with the hairpin loop your mRNA being produced from your DNA. Also, there are some introns and exons. Introns are the one which are being coded, whereas exons are being coded and introns are not. So they will be removed. So at the end you will have messenger RNA, including your exons part only. That we have discussed all these parts. Now comes to translation. This is the first stage of a protein biosynthesis. So in translation, produced by transcription is decoded by ribosomes to produce specific amino acid chains, polypeptides, that would later fold to have an active protein. So translation occurs in cytoplasms where the large and small subunits of ribosomes are bonded to the mRNA. So here you can see ribosomes are being attached to your messenger RNA, and then they are being producing with the help of a transfer RNA a long chain of a protein amino acids, mercury, glycine, leucine, valine, alanine, and they are being produced like this. So the ribosomes facilitates decoding by inducing the binding of tRNA with complementary anticodon sequence to mRNA. So tRNAs carry specific amino acids that are chained together into a polypeptide as a messenger RNA passes through and read by the ribosomes. So there is valine, alanine, glycine, and that's all. So entire ribosome's messenger complex will bind to the outer membrane of rough endoplasmic reticulum and release the nascent protein polypeptide inside for later vesicle transport and secretion outside the cell. Then there is a genetic code. So here on the left side you have to write. I have to keep checking the messages also so that, yeah, Pupinder or chickpea, I forgot that name. So yeah, all is good so far. Sorry students, I have to come back to see if the streaming is okay. So I keep stopping it to check if everything is fine. Okay, so then comes your first base, that is UCAG. Yeah, on the left side you write your UCAG, and on the middle, you second base, you write again UCAG, and then you write the four letters in this manner, and then you take U from here, U from here, and U from here. Then that will be your first UUU. Then comes your UUC, then UUA, then comes UUG. Like this, your phenylalanine, leucine, serine, tyrosine, cytosine, arginine, histidine, glutamine, proline, threonine, all your various patterns are being made like this. So what is genome? It's the entirety of organisms' hereditary information. It includes all the informations related to your genome of the heredity into this part. So it is encoded either in DNA or for many types of viruses in RNA. So genome includes both genes and non-coding sequence of the DNA. So comparative genome size being discussed here with the Homo sapiens, mouse, fruit fly, plant, roundworm, yeast, bacteria. So here you can see 3.2 billion size in this case, and gene number is around 25,000, and in the bacteria is 3,200. So like this, it's like, and the chromosome number 46, 48, 10, 12, 32, 1, 1, like this. So why genome analysis? The prediction of genes in uncharacterized genome sequence to obtain the complete sequence of as many as genomes as possible, and for genetic modifications. So genetic modifications develop a new variety at faster rate, like BT cotton, BT brinjal. So we can have better varieties if we are understood of the genome of one species. So now let's discuss. We have at the moment we are done with the basics. We are done with the basics of. But when Devikandan Kurmi, you have to increase the resolution of a screen or some problem at your end, because all of can see the screen actually. So if somebody cannot, just please try to check it. So this was the basics of your cell today, what we did, and basics of your molecular biology. Now I will touch the part of your basics of uh, molecular biology, that is tools are used in it. So I will not go in detail because that will be dealing in future, but at the moment we will touch the basics parts only. So gel electrophoresis, so we will deal this part in detailed manner. So gel electrophoresis, the basic principle is that DNA and RNA and proteins can be separated by the means of electric field. In agarose gel electrophoresis, DNA and RNA can be separated on the basis of size by running the DNA through an agarose gel. So proteins can be separated on the basis of size by using an STS page gel, or on the basis of size electric charge by using what is known as 2D gel. So as you can see, on the basis of electric charge, your DNA are being separated, and also basis on the size, and they're exposed to the film under the UV light, and then we can see the visible 500 base pairs DNA band here. Then there's a polymerase chain reaction. This is done to increase your amount of DNA in billions amount. So it allows single DNA sequence to be copied or altered in predetermined ways. So PC has many variations like reverse transcription PCR for amplifications of RNA, real-time PCRs which allow for quantitative measurements of DNA and RNA molecules. So PCR analysis. So here you can see your template DNA with your first cycle being producing 4 copies, then you have 8 copies, then 16 copies, then you have 32 copies. So this is how you have complete PCR, which will go exponentially and leads to the production of around 68 billion copies by the end of your production. And the primer is a strand of nucleic acid that serves as a starting point of DNA synthesis. So these primers are usually short, chemically synthesized oligonucleotides with a length of about 20 bases, and they are hybridized to a target DNA, which is then copied by the polymerase. So the minimal primer length used in the most application is 80 nucleotides, and application starts at the 3' end of the primer and copies the opposite end. So in most cases of natural DNA replication, the primer for DNA synthesis is short stranded of RNA. So what are the applications? They're used for the gene expressions pattern to understand them. Task of DNA sequences can also be assisted by PCR. PCR has also helps in the cloning, genetic mapping, also match for the closure to check the closure relatives, how they looks like, parental testing. So all these things are helpful with the help of PCR. Now comes your macromolecular blotting and probing. So this is like your southern blotting, northern blotting, where you take the genomic DNA in the southern blotting, then you cut them with the help of friction endonuclease enzymes, then you run over the agarose gel electrophoresis, and then you blot them on the nitrocellulose membrane, then you probe with the radio labeled phosphorus, then you can expose to the X-ray film, then you can have your whatever the bands that you're looking for, whatever the your things that you want to see, you can have it with the help of this technique. So southern blotting is a method of probing for presence of specific DNA sequence within DNA sample. So we can separate via the gel electrophoresis or via capillary action. Then membrane is exposed to labeled DNA probe that has complementary base sequence to sequence the DNA of interest. So southern blotting are still used for some applications such as measuring transgene copy numbers, transgenic mice, or gene knockout or embryonic stem cell lines. Then the northern blotting, it's the same thing, everything is same, it's just instead of DNA you have RNA being analyzed. And in the western blotting, instead of DNA and RNA you have protein analysis in this. So remember this S and W, S and W, southern, northern, western. So it goes for DNA, RNA and protein, DRP, S and W, DRP. So like this, you can remember this forever. S and P, so I will write this for you. S, southern, northern, western. This is for DNA, this is for RNA, and this is for protein. S and W, DRP. So remember this line, so you will never forget that which blotting meant for which blotting. And this is meant to be done with the help of primary antibody and secondary antibody. With the chemiluminescence and autoradiography. So molecular markers are based on naturally occurring polymorphisms in DNA. So genetic markers are sequences of DNA which have been traced to specific locations on chromosomes associated with particular traits. It can be described as a variation that can be observed. Then there are various genetic markers. Remember these full forms, they're always being asked in various net exams if you're studying. All is good. So RFLP is restriction fragment length polymorphism, AFLP, RAPD, BNTRs. So remember these terms, they are the various genetic markers which are being used to understand um, your variability. So five conditions that are set are suitable for molecular markers. That is, they must be polymorphic, codominant inheritance, randomly and frequently distributed, easy and cheap to detect, and it should be reproducible also. So molecular markers can be used for several different applications, including germplasm characterizations, genetic diagnostics, characterization of transformants, study of genome, organization and polygenic analysis, paternity testing, investigation of crimes. So all these things you can use for. So something about RFLP, it involves the fragmenting a sample of DNA by restriction enzymes, then recognizing that part with the help of your electrophoresis or any other technique. So the various advantages that they are variants and they are codominant. They measure variations at the level of DNA sequence, not protein sequence. And disadvantages is it requires relatively large amount of DNA. So AFLP, amplified fragment length polymorphisms. So in this analysis, we can amplify restriction fragments and reduces the complexity of materials to be analyzed. So it can be used for comparisons between closely related species only. So advantages are fast, relatively inexpensive, highly variable. So markers are dominant. Presence of a band could be individual, either homozygous or heterozygous. In RAPDs, they are in this type, we have PCR being used to amplify your DNA of the random thing. Then the microsatellites or SSRs or short tandem repeats are repeating sequence of 1 to 6 base of DNA. So they are highly variable, fast evolving, co-dominant, but they are quite expensive and time consuming also to develop. Then you have SNPs, they are the single nucleotide polymorphisms, like CG is being converted to TA like this. So we can change this in the crop and we can see a big change in our trait of our species. And there's a short random repeat STRs, they're in DNA, that leads to the production of two or more nucleotides, and a repeated sequence are directly adjacent to each other. The pattern can then range in the length from 2 to 16 base pairs and typically in the non-coding intron regions. Used in forensic cases, used for genetic fingerprinting of individuals. Then this is the principles of DNA, that is isolation and purification. So DNA can be isolated from any nucleated cells. So DNA is a giant anion in solution. So, so what are the sources of DNA? It includes blood, buccal cells, cultural cells, bacteria, biospecies, forensic samples. So what we do is DNA is being isolated to collect DNA from subsequent molecular analysis, and there are three basic steps in DNA extraction. So first is cell is disrupted. Your cell are being sonicated or grinded to remove the membrane lipids by adding a detergent also. Then your DNA is isolated by removing our proteins by adding a protease to it. Then precipitation is done using an ice cold ethanol or isopropanol. Since DNA is insoluble in these alcohols, it will be aggregated together, giving a palette upon a centrifugation. And this steps also removes your alcohol soluble salt. So this is your samples here looks like which has cell and protein degradation is being done. Then binding is done to your cells on that top. Then centrifugation is done. Then elution is done after the washing. Then you have pure genomic DNA afterwards. So what are the basic rules? That first you have to lyse the red blood cells with gentle detergent such as Triton X100. Then wash your cells that inhibits restriction enzymes. Then work on ice to slow down the enzymatic process. Then you can wear gloves to protect your samples from you. Then autoclave all the samples in this fridge and keep all pellets that you are looking for. So this is all the various, if you are preparing for NET, so these are the various things that you should be aware of. What type of cells are being used. NaCl is used to stabilize the DNA, remains as a double helix. EDTA, which is a chelating agents, magnesiums and cofactors which use the DNA rapidly. Anionic detergent SDS, which disrupt the lipid layers, helps to dissolve membranes and binds positive charge of chromosomal proteins to release the DNA into the solution. Then it includes protease to digest the protein incubator solution at an elevated temperature for 4 to 24 hours. Then get rid of the proteins, organ solvents extraction using equal volume phenol to chloroform 24:1. Then protein at the interface after centrifugation 10,000 rpm at 10 degrees Celsius for 10 minutes. Then you precipitate the DNA, add 2.5 to 3 volume of ice cold 95% ethanol to DNA, leave it for 20 degrees overnight, then centrifugate. Then you wash it with 70% ethanol, which is spent in there, sterilized DNA, and then store at 4 degrees Celsius. And this is the formula for quantifying the DNA. You calculate the optical density, multiply by the 100 dilution factor, and then 50 milligram per apple and divide it by that 1000. So if you're seeing this optical density be around 50 microgram per ml, then you have, at 260, you will have at single stranded RNA with 33. If it is RNA, then it's 40. If it is double stranded DNA, then it's 50 microgram. So as per the OD, we will have different types of DNA. And also we can check the purity of DNA. If the OD is coming out to be around less than 1.6, then a protein is contaminated. If it is more than 2, that chloroform phenol is contaminated. So in summary, you have sample of DNA extraction, you lyse them, put them in detergent, enzyme and salt buffer. Then you remove the cellular proteins and precipitate the nucleic acid with ethanol and quantitate the purity measurement of DNA. So you can use it for further agriculture development, environmental protections, and to ensure food security for growing human populations. So that's it for today from this part. So I will share these presentations with you. So about the virtual experience, we will do that tomorrow or not today because it will take me some time. So now your task is, if somebody have not understood, that you have to write you have to write one what do you say, one assignment which should include all the summary of what we have done today and send it on the concerned email ID, so that you can be aware of what you have learned the basics today. You have learned the various basics tools of molecular biology. You have learned the very basics of your molecular biology. You have learned the very basics of cell biology. You have learned the very basics of your transcription, translation, replication, how it is done. So all in all, I have brought you the taste of molecular biology and cellular biology in one go. So that was the main aim for first day class, the introductory class, so बच्चे, to know about exact volume and all these things, I have one protocol, I guess. It might be with me. Yeah, I have a lot of protocols. It's all from my PhD, so I can share some of them. So DNA extraction protocol. So this is the lysis of the cells. You do this way. Craniosus buffer, IPID protocol, heart crushing. Cardiomyocytes, SOP, Western blotting, English. So I am sharing with you some of my protocols. So go through them. Might be interesting for all of you. So you can send your assignments on this in the form of PDF or doc. on this email ID or on this email ID. Yeah. What else? I also have one book for all of you. It's called Principle of. It's a Wilson and Walker book. So I'm sharing one book for molecular biology and biochemistry. It's a very nice book by Wilson and Walker. So please go through that. Yeah, you can submit by tomorrow evening. Before tomorrow evening, before we start the class, you can send it at any time before we start with tomorrow. Yeah, all good. So I had shared with you some protocols that I had with me, um, and also Principles and Techniques of Biochemistry and Molecular Biology by Wilson and Walker. Very great book. Uh, it will be good for you. We used to study mainly from this book main only for our any topics that we were confused. That was the best one. Okay, students. If you have any other questions, you can write me on my what is this? Why am I forgetting so many things today? On my Telegram ID. So I try to reply. I tried it yesterday. So whenever I get time from my schedule, I try to reply them. So yeah. So all the best for tomorrow. So we are done for today's session. So tomorrow we will do with the cell biology, with the cell culture, animal cell culture, and one more thing, that is your microscope, various types of microscope and cell and animal cell culture. So these two things will be dealing with tomorrow's topic. Yeah, yeah, A4 size paper. Right, right. Okay, students, I got to go now. Thank you very much for today. You might be having another class soon, so join that class. So it was very nice seeing you all, teaching you the basics parts, and let's see tomorrow. See you tomorrow then. Take care. Bye bye.